On this page is a model of the histology of the liver. Now, like all the other models, this one enlarges, plays the rotation, enlarges, forwards, backwards. Now, I like this model because it gives you a very clear picture of what the liver actually looks like. So this is our liver histology slide. It's not that clear. So I can see some portal triads, 1, 2, 3, 4. There should be another one up here. And the central vein, but it's not the nice, clear structures that we should be seeing in a human liver. By the way, I think this is actually a pig's liver slide. So here's another shot. Here's a big vein in the center. There's like these 3 other things in the corners, but it's not really clear what we're looking at. So we're going to look at the histology slide model instead. So we're going to start with this side, and then we're going to look at this side, which is a close-up of that little section. The liver is arranged in these lobules, see the lobule, that are roughly 6-sided. So we have 1, 2, 3, 4, 5, 6. And in the center of these lobules is a central vein right there. That is called the hepatic. That's going to lead to the hepatic vein. So that's the central vein. And in the corners, there are these 3 vessels. So it's called a portal triad. So if we look at them, we see 3 vessels. If I just go all the way around, you can see the 3 vessels running all the way up. We have a branch of the hepatic artery. We have a branch of the hepatic portal vein. And then we have this duct right here, which will become part of the hepatic duct. So the 2 vessels, the hepatic portal vein and the hepatic artery, are taking blood in to be filtered by these lobules. And then waste products and whatever else the liver is producing is going to drain through the bile duct. So the liver is actually made up of these sinusoids, these canals. And they actually go all throughout. So we could see the hepatic portal veins actually go branch out, and we have the sinusoids all going here. And what's going to happen is these are sinusoid blood vessels, and blood gets filtered out and into. So the liver has these sinusoids here, and they go all throughout these lobules. So see the sinusoids there, and they're lined by hepatocytes. So these cells that are lining it are all hepatocytes right here. So these are all hepatocytes. And what happens is the plasma leaks out of the capillaries from the hepatic portal vein and the hepatic artery. And again, these are branches, not the main one. And they go through here to be processed by the hepatocytes. So the hepatocytes are going to be doing a lot. They're detoxifying things that were in the blood. They are going to be processing carbohydrates and amino acids, and eventually lipids too. They're going to be making blood proteins. They do a lot of things. So if we actually look at our labeled picture right here, so we have the sinusoids here, and those are the hepatocytes. We have the arterial from the hepatic artery coming in, so the plasma is going to go through here and be processed. Now, every once in a while, you're going to have these Kupffer cells. Those are macrophages. They're going to be having lunch on things that are not supposed to be there. Now, what happens is these hepatocytes are going to process things now as the plasma goes through. And the plasma is eventually going to reach the central vein right here, where it goes back into the blood and returns to circulate. So we can see all the sinusoids draining back into this vessel that's eventually going to become the hepatic vein. Now, what about waste products? What about the bile salts and bile acids that these hepatocytes are dealing with? Well, that is the job of the hepatic duct right there. Now, what's going to happen is there's actually a second canal in there. So if we zoom in on the canal, we're going to see there are these bile caniculi on the other side. And they're going to drain into the bile duct. And that is going to go into the hepatic ducts, and then it is going to go to the gallbladder. So that is our liver histology. It's actually a really cool model. And look at it. It'll help you with actually learning the liver histology, I think.